Hey everybody, my name is Bartuska, and I'm a rising junior at Oregon State University studying Oceanography and Climate Science. This summer, I've gotten to work in the Kiel-Reese lab, and I've had a lot of fun. To understand my project, you first need to understand the greenhouse effect and at least a little bit about the methane cycle. Greenhouse gases, such as carbon dioxide and methane, act as a blanket that trap UV radiation in the atmosphere. It's really important that we understand how much carbon is going into and out of the atmosphere so we can understand how much energy is being stored. That being said, there's about 4 billion tons of methane stored under the ice sheets and sediments in Antarctica. That's a lot of carbon that isn't currently being accounted for in the budget. My lab's research is understanding how that stored methane interacts with the environment. Microbes at depth form methane, and microbes in the upper sections of the sediment consume it in the presence of oxygen. This leads us to my research objective, determining the distribution of microbial cells in raw sea sediments. It's just the first step in understanding Antarctic methane exchange with the atmosphere. So before I got here, some of my lab mates got to go down to Antarctica and collect samples from the Ross Sea. I'm super jealous. Apparently the REU didn't have funding and the time frame didn't work out to send me with. My classes could have waited, but they didn't ask me. So when they were there, they collected jumbo gravity cores, like that one, to analyze the sediments within. When they got them on deck, they diluted the sediments at a 1:10 ratio with paraformaldehyde. Once I got here, I diluted them down even further, and then I filtered them onto black polycarbonate membranes and dyed them using cyber green. This is a fluorescent dye that makes DNA, genetic material, and cells glow green under the epifluorescent microscope that I used to count them. I found cell abundance to range from about 4 million to 100 million cells per cubic centimeter. There was negligible correlation between cell concentration and oxygen concentration, but something that I did find really interesting was that oxygen concentration was actually higher at depth than it was in upper sections of the sediment in most of the locations. That was just like an unexpected result that we still don't really know what the implications of it are, but we're going to find out. And then cell counts were higher at the surface than at depth in almost every single location that we sampled from. Like I said earlier, this is part of a larger project to recalculate the global carbon budget based on Antarctic methane. To do this, first, after my stuff, we'll perform bioinformatics, which will take DNA strands found in our samples and compare them to a known database, which will determine what kinds of bacteria, archaea, and other cells are present. Then, transcriptomics will be performed, which will determine the function of these cells, whether that's consuming methane, producing methane, having snowball fights at the bottom of the ocean, who knows what they're doing. Based on this, we'll know how many cells we have, what percentage of those cells are doing what kind of work. Using that, we'll be able to understand the carbon respiration, consumption, and production in Antarctica. This will give us a working model of raw sea carbon cycling, which can be included in the global carbon budget. This is really important because it's the kind of information that organizations such as the IPCC, the Intergovernmental Panel on Climate Change, use to inform governments on the implications and future risks of climate change, and it's how they advise them on mitigation strategies and policy decisions that will help protect our future and our planet against an ever-changing environment. Thank you.